Hello students, today's class we will have a recap of what we have done in the past, and when we will go into the uh, process of the development which we were covering in the last class. So developmental biology, the Wikipedia definition of the developmental biology is the study of the process by which the organisms grow and develop. So what is this growth and development? So the growth refers to the fact that from a single cell, that is the fertilized egg, the multicellular organism is formed with different types of cells which are specialized, and they form the complete organism. This is a process by which which keeps repeating again and again, time after time, whereas the development is a process that takes place throughout the life. So our body has billions of cells, and many cells die on each day, and the progenitors, um, such as the muscle or the bone marrow or the neurons, um, or the skin cells, have specific progenitors. So these progenitors produce these totally differentiated cells which take over the function of the dead cells. But if this developmental process is compromised, it might lead to the unlimited cell growth, which leads to the tumor formation. People believe that there is a paradox. That means there are not enough genes to encode the organism's complexity. And uh, it is known now that genes can be reused. So they may have specific timing, specific localization, they may have specific combinations of genes which work together in a specific dosage of a protein which is needed for doing that specific function. So the concept of one gene, one character is generally wrong, and this the typical example of this timing, localization, combinations, and dosage is, for example, plants. So plants do flower in a specific time of the year, some of the plants, and maybe these genes get expressed in that specific time, and they have a specific localization, and they work together with certain combination of genes which work together in order to flower in that specific time of the year. And probably they might require specific dosage of protein expression whereby these flowers can be seen in this part of the year in a specific plant type. So these cells also, there should be some kind of a self-organization as well. So the cells should talk to each other. There will be intercellular communication, so which is required in order to form the complex structures. Why this? For example, we have a bone. So bone is a different cell type. Osteoblasts make these bones, whereas also inside the bones reside the, the hematopoietic stem cells, which work in combination with these osteoblasts and the bones, and thereby they maintain the homeostasis of the body. So these are the complex structures which work together. There may be certain environment which need to be created inside the bone, so there are various cell types which could be involved in this process. Also, the best example of the genes being reused is, uh, for example, when a baby is born, at this particular point of time, the baby need to grow each week, or every 10 days, a specific body weight. So they grow up from half a kilogram to 1 kilogram in body weight every week or every 10 days. So what happens is, in the beginning is, there may be genes which are reused. Maybe there are oncogenes, proto-oncogenes, which may be used to produce large number of cells in order to increase the body weight, or the blood volume also has to increase, so there may be millions of blood cells which need to be produced quickly, and the blood volume increase, the body weight increase, so which has to take help of some of these genes. But these genes, when they grow into adults, have to be switched off. If they are not switched off, they may lead to the specific kinds of cancers. So people use different model systems, model organisms for various reasons. So there may be ethical reasons wherein the humans cannot be used as model organs to study certain disease or the developmental processes. Whereas these model organisms can grow very fast. So for example, so the Drosophila can be maintained in small tubes. You can multiply them whenever you want. They reproduce very fast, and frogs, for example, can give rise to many embryos. So these thousands of embryos can be used, so the experiments can be repeated in order to get the desired results. So these embryos can be utilized in the proper manner in order to get the picture of the development which the scientists wanted to study. There may be other organisms which can also be used as the model systems, such as fish, amphibians, and insects, because people believe that this development is a highly conserved process and this is an evolutionary process. So this picture depicts the neurulation stage where the neural tube is produced. So therein, people believe that this whole process of development can be highly evolutionarily conserved process. Like I said, uh, vertebrates, arthropods, mollusks, and even worms may have similar proteins and DNA sequences. And these genes and proteins from one organism can be used by the other organism. So some genes can be utilized across the species in order to uh, make the function normal of the other species. So the genomes of many of the model organisms are already sequenced now, and a lot of information is available. So there are these interesting, all these organisms, including mice or chicken in the early stages. Malfigi started his work with chicken eggs, wherein he studied the development. The people use zebrafish, they use the frog because they can give so many eggs, wherein the embryonic development can be easily studied. Salamander is a very good model system to study the regeneration process. Sea urchin was widely studied to know the regulative development, the theory of regulative development, and people used worms and flies as well, along with plants such as Arabidopsis. So if you look in the last class, we studied that the egg has two different hemispheres, the animal hemisphere and the vegetal hemisphere, and the you can see that there are different staining. The upper part, the animal hemisphere, is darker in color, whereas the vegetal hemisphere is lighter color. Also, we looked that there are dorsoventral and anterior posterior axis. So the anterior is related to the head region, whereas the posterior is related to the tail region. The dorsal means the back. Even in the human, it is the back side, and the ventral is the lower side or the front side in case of human. So also there is a proximal distal axis. The proximal axis means something which is closer to the body, and here in the humans also, this is closer to the body. Distal means something which is farther to the body, which is far away from the body, and lateral is the related to the side. So yesterday's class we saw uh, there are stages of embryonic development, and such as fertilization, oogenesis in the first stage, cleavage, and gastrulation and neurulation. So if you see the fertilization or oogenesis, And this stage, after the sperm enters inside the egg, there is egg activation which happens, and there is a formation of zygotic DNA. So that means the germ cells, which are the sperm and the egg, are haploid in nature, whereas the uh, zygote is diploid in nature. So there is a formation of zygotic DNA, and there is initiation of the developmental processes. So the symmetry is breaking. This is the symmetry breaking event. So during the cleavage, so the embryo divides in an extraordinary fast rate, and there will be cell division in each 30 to 40 minutes. E. coli multiplies at the rate of 20 minutes, whereas in this stage in the embryo cells divide at the rate of 30 to 40 minutes, and there is actually no growth in the size, but there is controlled, and this whole process is controlled by the maternal proteins or the mRNA. That means after the fertilization, there is no time for transcription or translation of the new zygotic genes. So whatever is present inside the egg, which is the pre-existing maternal proteins and mRNA, they control the gene expression and the transcription and the translation. And there is a creation of blastocele. That means the cleavage cavity, um, uh, and the blastula, which means the sphere of cells surrounding it. You can see here, this is the animal pole, and the cell starts to divide. There is a cleavage, 2 cells, and followed by 4 cell formation, and you can see that the cells start multiplying without growing in the size, and it fills up the zygote with multiple, it's a huge mass of cells. At this stage, you can see the blastula and the blastocele formation. And during the gastrulation, there are formation of 3 germ layers, which I mentioned. The father of embryology also showed that, uh, he identified that there are 3 layers in the chicken, and that there is ectoderm, endoderm, and the mesoderm. And ecto means the outer layer, which means outside our body we have the skin, and the mesoderm gives rise to the muscles, bones, and the blood vessels, whereas endoderm gives rise to the digestive tract, such as the stomach, liver, kidneys, and uh, there is a formation of the embryonic axis in this stage. And at this specific point, there is activation of zygotic genes, wherein there is a considerable movement of the cells within. So you can see here how the movement of the cells within the fertilized egg or the zygote is happening. You can see the movement, monitor the movement in these pictures, and you can see there is a blastopore lip, and gradually the blastocele is seen, whereas during the gastrulation, these cells starting to align themselves in a specific axis, and the cell starts to move. You can see these cells start to move in a specific fashion, aligning themselves um, in a specific manner, wherein you can see the red cell, the pink cell may give rise to a different cell types, and orange would give rise to another cell type, and the green and the gray can give rise to different cell types of the body. These cells start to organize in a specific manner, and they may be these cells might give rise to, the pink may give rise to head, whereas the orange may give rise to trunk, and green may give rise to the tail, and the bottommost, the 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 gray can give rise to the anus part. And there may be the endoderm may give rise to the cells related to the internal digestive organs. So you can see the place from where the sperm enters, opposite to is the dorsal side, and exactly opposite to it gives rise to the ventral side, and thereby there will be also anterior posterior formation, because of which all these cells starts to align and give rise to the specific organs. So this is the typical picture which I showed in the past, wherein you can see the animal pole, animal hemisphere has ectoderm, related cells, where outer layers cells are formed, whereas the vegetal hemisphere gives rise to the endoderm cells. Whereas the mesoderm, the middle part, give rise to the either blood cells or somites, which give rise to the uh, muscles, tendons, ligaments, bones, etc. And the dorsal part has the head and uh, the ventral part. Um, so this exactly opposite to the dorsal is the ventral part. So it was believed that there are fate maps. So these cells start to align the endoderm, mesoderm, and the ectoderm, gives rise to specific fate of cells. Now, during neurulation, the elongation of the embryo starts to happen, and there is a formation of neural tube and notochord in chordates, and there are also somites. These are the primitive cells which give rise to the muscles, tendons, and the bones, and it gives rise to the early organ predecessors such as kidney and heart. So these cells start to align in a specific manner in a dorsal view you can see here, and there are certain alignment which is followed, and they align themselves, which give based on which the cell or organ starts to develop in specific place. Further, there is a neural tube which starts to form, and this is how it looks like. But there was a landmark experiment by Spemann which showed that the which showed the outline of his experiments were that if we graft a tissue from one embryo to another embryo, and he wanted to see what happens if he does it, and he has his revolutionary idea was based upon cut and paste method. So he showed that if you do cut and paste, take a specific embryo part from one of the embryos and graft it onto the other embryo. And the main observation of this experiment was a graft of specific tissue, which he called as organizer, um, and to a specific location can induce the formation of Siamese twins which are connected by the belly. You can see here. So there is a complete new organism which is formed, and it is attached to the belly of the uh, of the of the other organism. And the conclusion of this is that embryo cells are not committed to certain fate. In the previous class, we studied the Rock's model of mosaic development, wherein when the frog embryos were half of them were killed during the four-cell stage, so only half of the other embryo was alive and half frog was formed. But this experiment shows that it is not true. So even a part of the embryo can give rise to a complete organism here. So, so the summary of the results are: a piece taken from the upper blastolip of a gastrulating amphibian embryo can exert an organizing effect on its environment. It is in such a manner that a piece can therefore be designated as organizer. So the piece of embryo which is taken from this blastopore lip can be an organizer and can impact in and provide a certain impact on its environment. Therefore, he called that as an organizer. So this is the original blastopore lip which was grafted to the bottom of another um, embryo. And these secondary embryonic primordia are always of mixed origin. So you can always take embryo from some other species, and then it can always exert the mixed origin, and there, because the two embryos are of different origin of different species, so an organizer of another species can be used for induction, and then a chimeric composition can be established with great accuracy and certainty. So the thing which was lacked in the Speamann's experiments were the controls and the statistics. So the authors do not provide any control experiments such as grafting to the other locations of the embryo and the other times of the embryonic development. And they also state that the development is impeded after the grafting procedure. And statistics, if you see from the Mangold's lab notebook, we can now see that only the 15% of the embryos survived the graft, whereas 85% of the embryos died. So spontaneous Siamese twin may occur in nature in true sense, but they may occur in a very, very low rate. So this is one of the typical example of the snake having two heads which are joined by together. So this is a typical example of Siamese twin. And so this uh, the Spemann innovation was that until 1923, an embryo had a predefined fate map where each layer, the ectoderm, endoderm, and mesoderm, can give rise to different cell types and thereby forming different parts of the organs of the body. The cells up to certain stage are pluripotent and can have many developmental phases. So this is what it shows, that cells up to a certain stage are pluripotent and can have many developmental phases, whereas the Spemann established the concept of organizer in this case. So that means the cells during in the embryo in a specific stage, the each cell has the ability to give rise to multiple cell types which are present in the body. And then there are concepts related to stem cells and which can control over the cell fate. So Spemann won the Nobel Prize in 1935, whereas the Hilde Mangold died in 1926. The Spemann organizer was found in all the vertebrates, including the humans, and the dorsoventral patterning has become a model system for embryonic patterning. And stem cells are the promise for the future therapies to replace the damaged organs. This is the summary